Hello and good morning, afternoon, or evening. My name is Stacy Tannenbaum and I am the lead of the Pharmacometrics Group in the US for Astellas Pharma Global Development. And I'm really happy to be with you to continue the presentations on model-informed drug development with Module 2. I'll be doing a review of the various model types commonly used for MIDD, and I've split this presentation into two parts to control the length. What I have here is my bio sketch. I'm not going to read it to you. It's here for you in the slides, but I do want to point out that at the bottom of the slide is my email address. If you have any comments or questions, please don't hesitate to reach out and send me an email. I'd be very happy to hear from the folks who are watching this presentation to get your feedback. So let me jump in and talk about why we do what we do. And when I say we, I mean modeling and simulation scientists or pharmacometricians. When a patient goes to the doctor's office and they're sick or they're not feeling well or they have a disease and they get a drug, all they really want to know is, when will I feel better? If I take this drug, when am I going to get a response? But the prescriber, patient, and regulator actually have many more questions. One is, what's the appropriate individual dose for a patient? If a patient doesn't respond or has an adverse event, how should the dose be adjusted? What happens if a patient misses a dose or takes too much? How soon will the effects start? And that's certainly a primary concern of patients. And how long will it last? In order for me and my team to be able to answer these questions, we have to understand all of the steps between dose and response. And we need to understand them in a quantitative way. We need to know what will happen if we change the dose, if we miss a dose, or if we take too much. And we do that using pharmacometrics. We do that using PK and PK/PD modeling. And I'll speak more about these concepts as we go through this presentation, but just in brief, PK relates the drug dose to the drug concentration profile. And when I say dose, I mean dose and regimen, dose and frequency, as well as the formulation. When I mention PD, that is how the exposure of the drug, whether it's the drug concentration, the area under the curve, the maximum, etc., leads to a downstream response. And that can happen either directly or that can happen after a series of steps and feedback loops. PD can be much more heterogeneous and complicated than PK, but we will touch on both of them today. Ultimately, why we do what we do is to get the right dose of the right drug to the right patient at the right time. And that can be as simple as this acetaminophen label, which is take 2 caplets every 6 hours while symptoms last for children and adolescents. For the case of acetaminophen, you have a fairly wide therapeutic range, which means most people will be covered by the same dose. However, sometimes you need to dial in a little bit more closely to that therapeutic range. And that's why in children, for example, children under 12, you might want to dose by body weight or by some other covariate that we'll talk about later that helps you to dial in on getting the appropriate individualized dose to a patient. But ultimately, my team works to help figure this out and make sure that we're getting the right dose and timing for each patient so that they get optimal efficacy and minimal safety issues. And how do we do what we do? We do it through the application of pharmacometrics. This is the science which deals with quantitative description of disease, drug effects, and variability. This is how we implement model-informed drug development. It requires proficiency in a number of areas, such as the biology of disease, PK and PD, as I mentioned before, and mathematics or statistics, as well as computational techniques. Pharmacometrics lies at the heart of what drug companies do, collecting data from animals, normal volunteers, and patients. And I would also add, we also collect data from literature on similar compounds and within that same indication. And then we quantify those data, we integrate it together, and we are able with that to determine what that data mean for optimizing drug efficacy and minimizing toxicity. And we do that by getting the dose right. I'm going to start with the simplest, just to make sure that we're all on the same page when we talk about modeling and simulation and defining what are models. So please indulge me if you are a mathematician, if you're very comfortable with models, just sit with me for a few minutes while we talk about this. At the very simplest form, models are mathematical relationships among relevant pieces of information. What I like to think of a model is a summary. It's a conceptual tool for translating complex, dirty, very heterogeneous real-world data into a simplified form, and that's a mathematical model. It's an equation. Yes, it generalizes the detail, so you're not going to see all of the small details that happen within each individual, but it gives us trends, and it allows us to summarize quite a lot of information into a succinct package. By using this, summarizing our information, data, and assumptions, we can then use them to probe alternate designs and outcomes. And that is simulation, and I'm going to talk a little bit more about simulation in a few minutes. So let's start with a model. Mathematical relationships among relevant pieces of information. And I'm going to start with the simplest model that we have, which is a linear regression. And you see these all the time. You see scatter plots, and you try to draw the best straight line that you can through that plot, and you end up with a dependent variable and an independent variable. And I'll talk about what those mean in pharmacokinetics and pharmacodynamics in a moment, but I just want to orient you to this plot. And you also have things like parameters. So for a linear regression, your parameters are the slope and the intercept. Now let's talk about this in the context of PK or PD. Your plot will probably not look like this, but hopefully you get the gist. So in pharmacokinetics, when we talk about a dependent variable, that is the drug concentration. The drug concentration is a function of the independent variables. Usually on the x-axis you'll see time, but sometimes you see dose or regimen. So these are the things that drive the dependent variable. And when we do our pharmacokinetic models, we get things like clearance and volume of distribution. We're able to estimate the half-life. We're able to determine the exposure, the maximum concentration Cmax, as well as the time that occurs Tmax. So these are the parameters of our pharmacokinetic models. And I'll be touching on all of these things again as I get into the modeling components a little bit later. For pharmacodynamics, the dependent variable may be something like a biomarker or a clinical endpoint, a surrogate endpoint, for example, or blood pressure. Sometimes these are yes/no. Pharmacodynamic variables tend to be much more heterogeneous than pharmacokinetic. Our independent variables are things like time and drug exposure. And our parameters can vary. As I said, pharmacodynamic models are quite a bit more variable, but generally we'll get some information on the minimum response, the maximum response, the rate of change of response as your exposure changes, and things like potency. And I will touch on these concepts in more detail further in the presentation. So I mentioned modeling and simulation. Now let's talk about simulation. And this is an application of modeling. And I like this quote in the bottom, and I'm afraid I don't know who to attribute it to, but I really like this. The ultimate goal of modeling is prediction, not description. Description is useful. It is important for us to be able to characterize our PK and PD that we've observed. But in order for us to optimize the dose, we need to expand beyond that. So what simulation is entering different inputs into our model to see what the output would look like, or how sensitive the output is to changes in certain inputs. And we'll talk about this in a little bit more detail further in the presentation, but you're really using a mathematical model to conduct experiments, and we call them in silico, as opposed to in vitro or in vivo. We can use simulation to optimize the design of future studies, and actually, we often try to optimize the design of our current studies as well, to estimate missing observations in the data and predicting observations outside of the data, to examine the sensitivity of the model output to non-controllable inputs, because that's very important. We may find out that we need to adjust our study design or get additional information to handle non-controllable inputs. And of course, to explore situations that you cannot do in the clinic. They would be ethically, financially, or physically impossible, but they provide valuable information. And we'll talk about this a little bit more when we get into the concept of clinical trial simulation. So during this presentation, I'm going to mention pharmacometrics as a catch-all term for several types of modeling, starting with the simplest, which is PK modeling. And as I mentioned before, that is how dose, becomes concentration and what is the impact of dose on our concentrations and exposures. We also have PK/PD modeling. This is really the simplest form where we look to see how the concentration either in the plasma or at the site of action begets the downstream response. We can actually look at modeling and simulation without any pharma involved at all. So we can look at disease progression modeling, and we can look at the disease and how it progresses over time without any kind of intervention. We can do that at a very, very simple level. Just look at the slope. Or we can look at it on a very, very, very micro level by looking at systems biology. And this is looking at the pathways of disease, feedback loops, and other complications to really understand how the disease impacts the entire body. But of course, we're in pharma, and so we want to talk about how the drug impacts both of these things. So we have the drug impacting these very simple disease progression models, and that's drug disease modeling. And quantitative systems pharmacology is how we link PK to these systems biology models. But let's start at the beginning. We're going to start with PK and PK/PD modeling. And that's because pharmacokinetics and pharmacodynamics contribute to many sections of the CTD in the package insert. And maybe I'm biased, but we provide foundational quantitative understanding of drug disposition and drug effects, both safety and efficacy. And if you look through the CTD, there aren't a lot of places where PK and PD don't touch. We help to support and inform key drug discovery and development decisions with PK and PK/PD modeling. So that's really where I'm going to spend the bulk of my presentation. I will touch on some of the other kinds of models that I'd mentioned in the previous slide, but what I'm going to focus in on part one is exclusively PK, and then during part two, we'll talk about some of the other types of modeling. So let us begin at the beginning and talk about pharmacokinetics. And pharmacokinetics is how the dose, regimen, and formulation leads to a concentration time profile. So let me start with the general principles of pharmacokinetics. It's often thought of as a mathematical process only, but really PK itself is a physical chemical process. The drug needs to get out of the dosage form into solution and be carried by active or passive measures from the site of administration to the systemic circulation. So that's absorption. It then needs to be distributed through the circulatory system to the site of action, as well as to the rest of the body. That's distribution. And this is why you sometimes see off-target effects, because the drug doesn't just go where we want it to go, unfortunately. The drug is then broken down by chemical processes, generally into inactive components. And then these inactive components, or the unchanged drug, needs to be excreted, usually by the kidney, by active or passive processes. To summarize and understand all of these processes, this is where the math comes in. We can simplify these to a set of parameters which quantitates the ADME of a drug. It gives us important insights into how much drug gets in and how quickly, how extensively the drug distributes out of the plasma, and how efficiently and quickly the drug is eliminated. And then other parameters which are derived from these give us useful information on how to optimally dose a drug. What's the optimal dose? What's the optimal frequency? That's things like the half-life of the drug, the exposure metrics like the area under the curve, the peak concentration or the minimum concentration, Cmin, and of course the time of the peak concentration. So now I'd like to spend a little bit of time talking about how PK is collected during the course of drug discovery and development. Then we'll finally get to how we actually analyze those data with models. But let's start with pharmacokinetics that's collected in preclinical studies. PK in preclinical is very, very useful because you can collect a really wide range of doses that you generally cannot do in humans. And that really allows us understanding of the edges of the PK processes. So we get to understand whether or not there's going to be saturation in any of our ADME processes. It gives us an idea of dose linearity with AUC and Cmax, and potentially when that starts to deviate. And the important part of PK in preclinical is that we can scale that to humans using a number of different ways. We'll talk on both allometric scaling and PBPK later in the presentation. PBPK is physiologically based pharmacokinetic modeling. PK/PD in preclinical is also quite useful in that we can get a really strong assessment of our exposure response profile due to the wide range of doses. And it gives us ideas of target engagement during different exposures of the drug. It allows us to assess the minimum anticipated biological effect level, MABEL, so the very lowest dose that we can give and see some kind of effect, as well as the no adverse effect level. And we can do that in order to help us identify exposure caps as well as exposure targets. So this will help us suggest safe and effective starting doses once we get into our first-in-human studies. Speaking of first-in-human studies, let's start there. And we're going to start with healthy volunteer studies. Single and multiple ascending dose. Some companies call it SAD/MAD studies. For the most part, I should take an aside to say that phase 1 studies are generally in healthy volunteers. In some therapeutic areas like oncology, it's done in cancer patients. And of course, that makes PK very, very difficult. You are also attending to things like variability, dose changes, dose interruptions, and other confounding factors like disease and standard of care. So we're going to put those aside for now. We're going to focus on the healthy volunteer part, as this is the most common and informative information that we have. When we collect these data, we have very rich sampling for PK characterization. And when I mention rich samples, I mean several samples per subject and generally covering the entire concentration time profile. So we get a really nice assessment of the PK in these patients. Again, just like in preclinical, this allows us to assess dose linearity within the doses that are used in humans. And if we, in our MAD studies, it allows us by looking at the PK at, let's say, day 1 and day 14, or when we think we're getting to steady state, gives us some idea of the steady state PK and the amount of accumulation that occurs with regular dosing. We can compare our maximum concentration to that safety cap that we mentioned in the previous presentation, or sorry, in the previous slide. And we can look at things like AUC and other exposure metrics to make sure that we are getting sufficient exposure for efficacy. This will help us in coming up with our later phase doses where we need to make sure that we're seeing appropriate efficacy. Helps us to understand the half-life of the drug in humans, which will help us to impact dosing frequency, as well as getting initial assessments of clearance in healthy volunteers, which will give us an initial assessment also of our variability. We'll plot clearance as a function of things like age and body weight to see whether or not we have very important covariates we need to investigate as we go into later phase studies. The primary PD that we collect in these studies is safety and tolerability, but sometimes efficacy biomarkers or other kinds of endpoints might be collected to help us with proof of pharmacology in some of the later studies. Let's talk about the clinical pharmacology studies. And these are a suite of studies that are used to have preliminary understanding of external factors on drug PK, usually in healthy volunteers again. So these are things like the food effect study, renal and hepatic impairment studies. Sometimes, depending on the type of drug, we might look at age and gender, like elderly versus non-elderly or children versus adults for age, just to be able to understand whether or not these are important covariates downstream. We do mass balance studies. Bioavailability is important if we're not going to be using an initial IV dosage form. If we're going into oral dosing or subcutaneous, you have to understand how much drug will get into the system. Of course, there's a thorough QT study where we look at the QT as a function of drug concentration. And then there's drug-drug interaction studies. And some of these studies, due to the fact that we have things like physiologically based PK models, some of these studies in well-understood indications and well-understood pathways can be supplemented or even replaced by PBPK. And we'll speak on that when we get to the PBPK sections shortly. Now, as we move into the later phase studies, we start to look at the PK in our target population. The goals for PK in these particular studies are to understand and quantify the sources of inter-subject and inter-occasion variability in our target population, and to address whether or not this variability has an impact on dosing and exposure. But PK characterization in later phase studies can be a lot more complex because it's generally very sparse sampling. You may only have maybe 1 or 2 samples per subject, and it's not necessarily going to be at the consistent times that you'll see in these SAD/MAD studies. And you have more missing data. Patients drop out, patients discontinue or change their dosing. And speaking of that, we often have incomplete dosing information. For the most part, these later phase studies The patients are coming in for scheduled visits, but they're taking their drug at home. So we have to make some assumptions around the times and dates that they're taking the doses if these aren't things that are being routinely collected and written down. So there is some incomplete dosing information that we need to contend with. And there's also a lot more intrinsic and extrinsic factors to consider. There's a lot more heterogeneity in a patient population, other medications, for example, and other standard of care, as well as disease progression that we need to take into account for this patient population that maybe we didn't need to consider for healthy volunteers. And so as a result, there's going to be higher variability. But we have methods to be able to assess all of these things, and we often will pool our data, if appropriate, with some of our richer PK studies so that it can help to supplement the information that we're missing with these other patients. So now finally, we will get to the meat of this presentation, which is to look at some of the methods and models that we use for PK analysis. I just want to clarify, you often hear people talk about PK analysis in two different ways. The term is used both in bioanalysis as well as in pharmacometrics. So what I'm going to be talking about today is not the extraction of the drug concentrations from blood samples, but the quantitative methods for determining the PK parameters, like we talked about before, clearance, volume, and half-life. So now let's proceed to talking about PK models. And I'm going to start actually by not talking about modeling, but talking about the quantitative analysis of PK data, and that is non-compartmental analysis. So in a case like this, it's usually done in early phase studies with rich PK. So we have a few subjects, but we have lots and lots of samples per subject, richly sampled data. And in non-compartmental analysis, you only need a piece of paper, you need a piece of graph paper and a ruler and a pencil and a calculator. So you don't actually need to have these really strong computational methods to be able to determine the PK in individual patients. We extract their information directly from each individual's data. So for example, we get things like the area under the concentration time curve. So this is a PK profile for an individual patient. It's obviously simulated, but the area under the curve is essentially we can calculate using something called the trapezoidal rule, or we can calculate directly the area under that curve. That gives us an estimate of the exposure of that patient, in this case over 24 hours. We can also get the Cmax and the Tmax, which we've already defined, but it's the maximum concentration as well as the time at which that occurs. And we can get things like the elimination rate constant, which is the line that you see here on the plot that just appeared, as well as, to some extent, the absorption rate constant if you do something that's called curve stripping. From those, we can derive parameters like the half-life, the clearance, the volume of distribution, and if we have both IV and oral or subcutaneous data or non-IV data, we should be able to also determine the bioavailability of the drug. Now, non-compartmental analysis is easy. As I mentioned, you don't even need a computer. And it makes very few assumptions. Because there's no underlying model, we just take each subject's data as it comes, and we take that as the whole truth. So it doesn't really help us to understand the variability in those patients, but we take each patient as the whole data set. Automation is still possible using appropriate software, so you don't have to do it with graph paper and a pen. And the nice thing about non-compartmental analysis is because it's easy, it'll also give us some assessment of variability and accumulation. So this is the situation that I mentioned before, where if you have day 1 PK compared to, let's say, day 14 or some steady state PK, it'll give us an idea of the accumulation in patients and variability across patients. The disadvantages, though, is that it does require many samples because you're really counting on the data to give you back those variables that you need. So it's highly dependent on things like sampling time. Because you're pulling Cmax and Tmax right off of the plot, you need to make sure that you're collecting them at the appropriate time. If you miss it by an hour on either side, your Cmax and your Tmax pulled off the plot will not be the true Cmax and Tmax. And you have to have sufficient samples to define the terminal phase as well. Generally, you want to have 3 to 5 samples in the terminal phase to get a true assessment of the half-life. And it tells you nothing about the mechanism. You can't determine if there is nonlinearities or complex absorption. You really just have to take the data at face value. And you have little predictive value. You can't interpolate or extrapolate from non-compartmental analysis because you're taking each individual's PK as it is. So in order to be able to circumvent some of these disadvantages, generally we turn to compartmental PK. And I'm going to start talking about compartmental PK applied to individuals. So this would be a situation where you still have few subjects with lots of samples per subject, and you're fitting each subject one at a time. Now, unlike with non-compartmental analysis, where you got things like AUC, the elimination rate constant, and Cmax and Tmax off the plot, in this case, they're derived variables. So we start with our parameters from the model. So in the model, we estimate the clearance, volume of distribution, the elimination rate constant, and then again, depending on whether or not we have IV and oral data, the bioavailability and the absorption rate constant. And then we derive the parameters that we would normally take off of the plot in non-compartmental analysis, like AUC, Cmax, and Tmax. So let's talk a little bit about the advantages of doing compartmental analysis versus non-compartmental. And I alluded to these before. One advantage is that you do get more ways to understand PK. You have more parameters like absorption that you can actually put functional forms in and understand the absorption a little bit more deeply. You don't have to do curve stripping, assuming a very specific kind of absorption. It can fill in sampling gaps because you have a model for each subject. You can do interpolation and sometimes extrapolation, which I'll speak to on a future slide. It gives us, again, the initial assessment of variability, similarly to the way that it's done with NCA, where you can look at clearance for each patient as a function of things like age and body weight and determine the variability across those patients. And if you do have nonlinearity, it's okay. If the functional form is understood, and we often do have a functional form for saturation and things like that, we can work those into our models. And so that helps us to assess the nonlinearity as well. The problem with compartmental PK is it is modeling, so it requires specialist software and training. And sometimes as you're starting to add more parameters because you're starting to try to assess more things, some parameters might not be identifiable. If you don't have enough data to be able to determine some of these parameters, you may not be able to assess them properly. And it's model dependent. Obviously, it's compartmental modeling. So you are assuming that the same model is applied to all individuals, one size fits all. And for the most part, that's generally true, but there are always exceptions to the rule that make it difficult for us to work around that. And just like in non-compartmental analysis, there is some dependence on sampling. For example, if you want to get Cmax and Tmax, it is important to try to sample around the time that normally would happen. So let's talk about a couple of different compartmental models that you will see very commonly. And one is the one compartment model. So one compartment model is essentially a bucket with a hole in it. So you administer the drug to the patient. It is immediately mixed and all over the body. And then you have elimination from that bucket, from the person. And so it's very, very simple if you plot the concentration time profile on a semi-log plot. So the y-axis is on a log plot. One compartment model is just a straight line. And you'll see the same thing when you have oral dosing, where once the drug is absorbed, so there's an absorption component, you'll see this straight line. Surprisingly, well, actually not surprisingly, this is a fairly common way of understanding PK in patients, and it works in a lot of different cases. But probably what's going to be more palatable to most people and more understandable to most people is the two compartment model, because this gives the drug some time to distribute amongst the circulatory system, and into the tissues and the organs. So before administration, what you can see on the left side, there's no drug. After administration, it goes into the central compartment, which generally contains the heart, the lungs, and the eliminating organs like the liver and the kidney. And then after a time, the drug gets distributed throughout the system. And what you'll see is this bi-exponential plot. You'll see this sort of L-shaped curve, for lack of a better term, where you have in the beginning a very, very fast drop in the drug, in the concentration in the plasma, because you have distribution happening at the same time as elimination. So drug is leaving the plasma into this second compartment, into the tissues, and then over time, only elimination is occurring because there's no more distribution. Your body's in equilibrium. So these are fairly simple models, but they are really quite useful and really quite common. We do occasionally move to a third compartment, but generally the 1 and the 2 compartment models are the most common models that we look at in PK. Now, what can we do with these? Well, the utility of compartmental models is that we can do things like interpolation and extrapolation. So if this is one subject and that line represents this individual's model, and I took a PD sample at 4 and 16 hours, and I want to know what was the concentration at that time, we can pick those off the curve. And similarly for extrapolation, if I have this curve for this patient, I can extend that curve out to understand what happens after I've stopped sampling. So interpolation and extrapolation are both, these are forms of simulation, and they're very, very, very useful for us to be able to fill in gaps. And in particular, like in the interpolation component that I mentioned, to be able to match our PK and PD at the same times is always quite useful. So if we take the same patient PK profile, and we want to know, instead of interpolating or extrapolating, we want to know what would happen if we halved the dose or doubled the dose or gave it half as often or twice as often. We can do that because this purple line that's going through these points is actually representative of an equation that represents this patient's PK profile as a function of dose and time. So if we wanted to see what this patient would look like at half the dose, at 50 milligrams instead of 100, or at 2 times the dose, we can do that by plugging in different doses into the equation. And similarly, let's say that we're concerned about this patient's maximum concentration. They're doing well on efficacy. We want to get the same exposure. We want to get the same dose into these patients over the course of a day, but maybe we want to lower this patient's Cmax. So we can do that by dividing the dose or by giving it, let's say, 4 times a day, where you can see that the maximum concentration is reducing, whereas the overall amount of drug the patient is getting in a day doesn't change. So these are some of the things that you can do with these compartmental models that allow us to really help to optimize the dose in patients. So in the case of compartmental PK applied to individuals, I think you can see the utility of it, but when we're applying compartmental PK to one subject at a time, you really need to be in a situation where you have few subjects and many samples per subject. When we talk about compartmental PK applied to a population and its individuals, that's a situation where you often have many subjects, sometimes many, many subjects. So we're talking about as we're getting into some of our late phase studies, and we can have hundreds, if not thousands of subjects, and very few samples per subject. So you have very sparse data. Parameters that you get from the model are the same as that you would get from the compartmental model applied to individuals, except one of the really useful things that you can get is understanding the impact of covariates and quantifying the impact of those covariates. And I'll describe what I mean by covariates in just a moment. Why is this important? Well, we know that if you give different patients the same dose of a drug, you're going to get some slightly different, if not fundamentally different, PK profiles. And that is because patients, in particular, as you get into the later phase studies and you have much more heterogeneity, you have a lot of subject characteristics that could impact the PK. And I've alluded to some of these before: age, sex, and body size. But as you move into patients as well, you'll have things like concomitant medications. You'll have different markers of health. Could be disease status or renal or hepatic function. You could have pharmacogenomics. For example, I am a very, very slow metabolizer of 2D6. I can drink an entire bottle of codeine and nothing will happen to me. I will feel no effect at all. Whereas I remember a friend of mine said she once drank one dose of codeine and ended up on the floor for 6 hours. Very, very fast metabolizer. So we can incorporate those things into our models. Other things like diet, smoking, exercise, and standard of care can also impact the PK. Some of these things we can measure, some of these things we can't. But why is this important is that different exposure can beget different responses. So it's really up to us to be able to quantify why patients have different exposures, and if they are so different, do we need to modify the dose? And ultimately, that is the ultimate question that we have to try to answer. So unsurprisingly, we answer the questions using modeling. And in this case, it's nonlinear mixed effects modeling, otherwise known as population PK, sometimes POP-PK. This is a model-based approach of analyzing data from all the individual subjects together that helps us account for how individuals differ from each other and how the overall population behaves. So we're using the same compartment models that we talked about before, the one compartment model, the two compartment model, but we're adding on a variance model that helps us to quantify the intersubject and the inter-occasion variability, and I'll speak to both of those. Let's talk a little bit about population PK and what are its advantages and disadvantages. Well, what's nice about POP-PK is that you can use rich and or sparse sampling from subjects. Subjects borrow information from each other because you look at the population as a whole, you pool all the data together, if you will, to get a trend for the entire population. So that helps us to get a feel for what the overall underlying model looks like. And then we can start applying that model to subjects who only have sparse data. And as a result, we also have methods within population PK for handling missing data within subjects. It gives us both individual information, so you might hear them called post hocs or EBEs, but all these are individual PK parameters, so an individual's clearance and volume, as well as population parameters, which sometimes we call typical values of things like clearance and volume. And of course, there's more parameters, so it helps us to quantify variability and the impact of covariates on our PK. But it is complicated, and so it requires even more specialist training and software than even the regular compartmental modeling does. And because we have even more parameters to fit, just like with compartment modeling, we're now adding on even more. So if you don't have sufficient data, you might have some parameters that are not identifiable. Similar to what I spoke about before, it is dependent on the model. Generally, you are applying the same model to all individuals, although within Non-MEM, which is the software that most of us use for population PK, there are ways around this, such as mixture models, where you can propose two different models and let the software decide which model each individual belongs to. And to some extent, it's sampling dependent. So if you can borrow information from other people, but if you've sampled all of your patients in the initial stage of your PK profile and you don't have anything in the terminal phase, you're not going to be able to get good model characterization. As I mentioned in a previous slide, we often will pool together our rich and our sparse PK in order to have more support for the characterization of PK in our target population. To orient you a little bit to how population PK works, and this is really as technical as it's going to get, these are two different subjects. You have the subject that's in gray and you have the subject that's in black. And the solid line that goes through the middle is the population mean. So if we didn't have those two small lines going through the individual patients, this is the best straight line that you can fit through these two patients. And so this is a straight line fit. Similar to what you have before, we have a slope and an intercept. And so you have these typical values for the intercept and the slope, which are going to be your population values of intercept and slope. Then when you look at each of the individuals, what you get is an intercept for each subject and a slope for each subject. But rather than fitting them individually, you use that population model, the population values of intercept and slope, and you just adjust them. So you have these little U parameters that you see here that are the adjustment for the intercept and the slope for each individual patient. So then you have these, that's how you get the individual patient values, or those post hoc values that I mentioned before. So this is where we talk about intersubject variability. And remember these values when I talk about these UIs. We're going to talk about them again in a minute on how it helps us to assess the impact of covariates on our PK. And last but not least, we always look for each individual and how far they veer from their predicted line. And so that's residual variability. And so we add on this variability for each of our patients. Now, I don't expect you to remember all of this, but just generally to get the principle of this is we start with the population, we get the best model for the population, and then for each individual, using that population model as a foundation, we then find the individual parameters that will make them fit appropriately. hopefully that was clear. That's as much technical stuff as I'm going to go into, but if it's not clear, please feel free to send me an email and I can try to explain this a little further or provide you some references that will help you understand POP-PK better. The important part of POP-PK is one of the things that it gives us is the ability to quantify covariate effects. So those UIs that I was talking about before, those are sometimes called ETAs. These are the differences from, in this case, the typical value of clearance. And what you can see is that when we plot these versus two different covariates, in this case it's alpha-1 glycoprotein, which is a plasma protein, and on the right side is fat-free mass. And what you can see here is that there is a relationship. People with a high AGP tend to have a lower clearance. People with a low AGP tend to have a higher clearance. So because we know this, we can then put these components into the models. So if you take a look at the model on the right side, if somebody were to walk into the clinic, I would assign everybody the same clearance, which is why you're going to see these errors. However, if I put AGP and fat-free mass into the model, then when someone walks in based on their AGP and based on their fat-free mass, I can get a much better assessment of their individual values of clearance. So I start to reduce that error. And that helps us to refine each individual's needs. So if you have someone who has a very high clearance, that means they're getting rid of the drug faster. We may need to give them a higher dose. If you have someone with a very low clearance, they're getting rid of the drug slower, we might need to give them a lower dose. And so by understanding the impact of covariates and understanding how extensive these relationships are, that can help us decide whether or not we need to put covariates into the label. And I already showed you a label in the beginning for acetaminophen where we had weight in the label for children because weight is such an important covariate. And similarly, covariates can impact dosing. This Southwest Oncology group is quite old, it's from 2001, but you will often see milligram per kilogram dosing or milligram per meter squared dosing, particularly in oncology. And on the right-hand side is the Xolair dosing table, and in this case, it actually is dependent upon two covariates, your IgE level when you enter the clinic, as well as your body weight. So in these particular cases, they may have a narrow therapeutic range, or we need to make sure that we are dosing in order to make sure that the patient is getting the optimal safety and efficacy. And as a result, we'll put the covariates into the label. So up until now, most of the models I've been talking about, like the 1 and 2 compartment models, have implied that we have linear pharmacokinetics or first order pharmacokinetics. I've alluded to the concepts of nonlinearity and saturation, but I'd just like to spend a little bit of time talking about that because we have to incorporate those things into our models. You'll sometimes hear linear pharmacokinetics referred to as dose-independent, dose-proportional, or first-order pharmacokinetics. But these three components, that the PK parameters are dose-independent, that if you divide your concentration by dose and then you plot concentration time profiles on the same plot, they should be superimposable, or that the exposure, like the AUC or the Cmax, is proportional to the amount of available drug. Linear pharmacokinetics essentially means if you double the dose, you double the concentration, and you double the exposure. So generally, if we're going to change the dosing regimen, change the amount, change the frequency, this leads to predictable concentration profiles. And the interpolation and extrapolation figures that I showed you before all assumed linear pharmacokinetics. However, particularly at high doses, it is possible to saturate one or more of the PK processes. So for example, if you have active transport, such as transport into the gut or PGP, which is sort of the goalie to keep drugs from passing into the system, or you have active secretion, active reabsorption in the kidney, these things can be saturated at high doses because it's capacity limited. Similarly to protein binding, you only have a certain amount of protein. So when the doses are very high, you've saturated all of your available protein for binding. This can also happen when you have time-dependent PK, such as up or down regulation of an enzyme, as well as target-mediated drug disposition. And if you haven't seen this episode of I Love Lucy, I think this does a very nice job of explaining how saturation works. But what happens with nonlinear PK is that changes in dose or dose regimen can lead to a disproportionate increase or decrease in the plasma drug concentration or the exposure. And it's not always predictable. So it can be a concern if a drug has a narrow therapeutic window. If you think about this, if you've just shifted the dose a little and you expect then the concentration to shift a little, but it shifts a lot, it goes up quite a bit higher, that can push you outside of the therapeutic window. So we have to be aware of the nonlinearities in PK and be prepared to account for them. And of course, the way that we account for them is through modeling. There is an equation called the Michaelis-Menten equation, and I'm showing this to you to familiarize you with this functional form because you're probably going to see it as a way to capture potentially saturable processes. So what this is the rate of some kind of process, and generally we use it for clearance. The rate of elimination is Vmax, which is the maximum rate, times C, which is the concentration, over Km plus C. And Km is the concentration at which the rate of the process is half Vmax. If you've ever seen Emax models before, this is going to look very familiar to you, and you'll see Emax models a little bit later. It has the same functional form. But what's important to look at is not so much the curvy part, but really the two ends. Most drugs undergo saturable PK at high enough doses. You just might not see it at clinical doses. Where you're more likely to see saturation is when you are in the preclinical stage, when you are pushing the doses as high as possible. And that's really important for us to start to understand these and see whether or not we have to be concerned about this as we get into the clinical dosing. More likely what you're going to see at clinical doses is where your drug concentrations are quite low and much lower than Km. And what you can see is that red line that's inside the box is a pretty straight line. So what you see is a proportional first order PK there. So it reduces to first order PK when you have a linear relationship between the elimination rate and the concentration. So it's not that you don't have saturable PK at all, it's just more likely at the clinical doses and the clinical exposures that we're seeing, we are not actually seeing these saturable processes. But the reason I'm showing this to you is just to make you aware that we do have functional forms to help us account for these nonlinearities in PK that you might see. One of the places where you're very likely to see nonlinearities as well as other complex modeling challenges is when it comes to absorption modeling. You know, I mentioned on several occasions that one compartment and two compartment models, for the most part, represent the distribution of the drug within the system, so the DME of ADME, but the absorption piece can be incredibly complicated, and that's because there's so many formulations and so many ways to get the drug into the body. Intravenous is obviously going to be the most simple. That goes straight into your systemic circulation where you don't have to worry about absorption. But even with oral dosing, it can be extremely complicated. Part of the reason that drug absorption, particularly oral absorption, is so complex is that it's dependent upon not just the dosage form itself and the physical chemical characteristics, but also the anatomy and physiology of the drug absorption site. So many potential complexities, including gut pH, which can impact how the drug goes into solution and how it dissolves, gut motility and carrying it to the site of absorption. We also have active and passive transport. Passive transport just goes through a gradient, so that's not really dose dependent, but active transport, such as being pulled by transporters across the gut wall into the portal vein or into the system, or PGP mediated efflux, can be saturated, as I mentioned on a previous slide. And then, of course, there's metabolic complications. We often will lose drug from our dosage form due to gut wall metabolism as well as first pass metabolism, which can cause all kinds of complexities. So a lot of times what you're going to see if you're reviewing modeling reports, you may see that the PK looks pretty good after the absorption phase, but what you're going to see is a lot of model misspecification during the absorption phase. And that's just because of these complexities. So we have a lot of different models that we can use for absorption, and I'm not going to go through them in this presentation, but we have first order absorption, zero order absorption, a combination of the two. We have complex models that can handle lag times, that handle gradual absorption, which is called the transit model, and other nonlinearities. And many of these are good enough. They're empirical. They suit our purposes, and they do good enough for us to be able to characterize the PK well enough. But if we really need to fully understand the absorption process, because there is so much physical and chemical and anatomical components to it, and we want to ensure that we're getting adequate drug exposure in the systemic circulation, it may be necessary to turn to a slightly more complex but physiologically realistic type of modeling, and that's called PBPK, or physiologically based PK. So the next section I'm going to talk about focuses on PBPK. So when we talk about the 1 and the 2 compartment models that we talked about before, we tend to call those empirical models. Each of the compartments is not necessarily representative of organs and tissues, unlike PBPK. So in PBPK, this is a concept that provides a mechanistic approach to study and predict the pharmacokinetics of drugs. And it's really based more on physiologic and anatomic characteristics, like some of the ones I talked about on the previous slide, as well as the physical and chemical properties of a given drug. So I'll go into those in a little bit more detail. But because of the fact that it is physiologically, chemically, and anatomically based, it's really quite ubiquitous. It's really used probably the most across the MIDD3 landscape. So it's used in formulation development to help to predict the absorption of a compound, and can also include the food effect studies and other studies in the GI tract. There's preclinical to clinical scaling. You can develop a PBPK model in a rat and scale it up to humans, for example. There's drug-drug interaction studies. This is an extremely common use of PBPK, particularly in the cytochrome system, where we really understand the mechanism of action and we really understand the impact of certain medications on cytochromes, so we can predict DDIs in many, many drugs. Also in healthy to disease scaling, so you can go from healthy adults to, for example, renal insufficiency or hepatic insufficiency. And it's used very commonly also in adults to pediatric patients. So PBPK is worth understanding and knowing a little bit about, because you're going to see it in a lot of different places in the CTD and in making some of the drug development and discovery decisions. So this is a PBPK model. I know it looks scary, but really it's actually much more palatable to people than usually these more empirical models that I showed you before, because each of these boxes represents an organ, and each of these lines, these arrows, represent a blood flow. So these are the organism-specific parameters. There's things you can weigh, there are things you can measure. Each of these boxes represents a real thing, and you have things like organ volumes, blood flows, hematocrit levels, the composition of organs, gastric emptying time. These are things that are specific to an organism, and usually you can measure or you can weigh. Then there's the drug-specific parameters. But when I say drug, I don't mean this drug. I mean this. I mean the chemical compound. And these are things like lipophilicity, molecular weight, acid-base class, pKa. I mentioned a couple others previously. And so those aren't going to change when you go between different organisms. The chemical compound is the same. What you do need to try to make sure you assess is the drug and organism specific parameters. So what is the unbound fraction of the drug in the plasma? And that's going to depend on plasma protein levels, and how tightly it binds to the plasma proteins, for example. Things like blood to plasma ratio of the drug, tissue plasma partition coefficients, intestinal permeability, etc. So these are things that you would have to be able to assess for your particular organism and for the compound. Now, what's neat about PBPK is that it can kind of expand or contract depending on the level of granularity that you need. So in this particular case, here's a model that looks at absorption on a very, very micro level. You can see each of the components of the small intestine, the duodenum, the jejunum, etc., all the way through the colon. And you actually have different compartments for drug that is released, drug that is dissolved, and then drug that goes into the systemic absorption. So if you need to understand it on a granular level like that, you can do that. But at the same time, you can also contract. So this is a situation where you have a rest of body compartment. So you have organs and tissues that are of interest for the particular drug, or things that you can measure or things that you can weigh, but sometimes it doesn't really matter where the rest of the drug goes. It's like in a mass balance where that's essentially all the remainder of the mass balance. So I think what's nice about PBPK is that you can expand it or contract it based upon your needs. So I'm going to walk you through a PBPK workflow, and these slides are courtesy of Andrea Edginton at the University of Waterloo. She's really an excellent lecturer on PBPK, and I borrowed this schematic from her. And in the schematic, we're talking about building an adult PBPK model and then scaling it to children. But this could really be the same if you were building a rat model and scaling it to humans, or building a healthy volunteer model and scaling it to disease patients. So we're going to use adult as our reference population. And what you do in PBPK is you build the model, and then you predict or you simulate the adult plasma PK profile. One thing that's important to note about PBPK is that there's no human data yet. This entire model is built on first principles. It's called bottom up. So we have chemistry, physiology, pharmacology, in vitro data. These are things that you use to build the model, and then on top. So once you have collected some data, you overlay the observed data on top of our prediction. And we look to see, does the model adequately reflect the observed data? If it doesn't, and in this case it doesn't, we optimize parameters to match the observed and simulated profile. So this is called model calibration. Now, I want to be clear that when we talk about optimizing the parameters, this is not just, hey, let's double the intrinsic clearance and see what happens. It has to be built on rational, physiological, pharmacological, and realistic assumptions. And so you have to justify these changes when you make them to ensure that you're properly characterizing what's really going on in the system, at least to the best of your knowledge. So you're basing this on your understanding of the pharmacology and physiology of this drug. And then once the model is calibrated to your satisfaction, you then have your final model in adults. And this is where the beauty of PBPK really comes in. Because now that you have the final model in this population, you can then scale it to another population. And that's what we're going to talk about now. So as I mentioned before, we're using adults as our reference population and children as our scaled or our new population. But again, the principles I'm about to talk about will apply whether you're scaling from a healthy volunteer to a sick patient, from rats to humans, etc. To scale adults to children, so we'll use that as an example, you would scale things like protein binding. Children have different levels of protein available to bind to the drug. You scale clearance. Children have smaller kidneys, smaller livers, and they're also just not as mature in their pharmacokinetic processes. And there's actually an entire field of study called ontogeny that looks to understand how children from birth all the way through generally 2 years old, where they're considered to be roughly equivalent to adults in terms of the maturity of their pharmacokinetic processes. But a lot of these equations are based on age or body weight, and so they will change as a function of time. And of course, you scale anatomy and physiology. Children are smaller, so they will have smaller kidneys, smaller livers, smaller organs. Their volumes and blood flows will change accordingly. So when you scale this adult model to children, or from your test or your reference population to your test population, if you have data, then you can evaluate your predictions against it. If you don't, then that would be a situation where you just put a lot of uncertainty around your predictions. But in a lot of cases, a lot of these scaling parameters are well understood. If you have data, though, you can calibrate or optimize as appropriate to come to your final model in children or in your test population. And this will give you an opportunity then to probe that model for different questions that you have to answer in terms of, let's say, dosing. In particular for pediatric patients, we'll often use PBPK modeling to do exposure matching so that we can come up with the pediatric dose to have the same exposure as you do in adults. Now, as I mentioned, going from adults to pediatric patients, there are ways to do this without PBPK. The most common way to do so is allometric scaling. It has been found that many processes and rates scale across and within species, usually to the power of 0.75 for clearance and the power of 1 for volume of distribution. So if you plot the rate of elimination versus body weight on a log-log scale, you will actually see that all the way from a very, very small to a very, very large population, you roughly get a straight line. And so allometric scaling is often used when you're going from adults to peds, or when you have a very, very wide range of body weights within your adult population, in order to be able to address the fact that larger people have larger organs and therefore have larger clearances. So we account for these using allometric scaling. This isn't a PBPK principle, but I did want to make this point before we leave the topic of pharmacokinetics. So part one today focused on PK principles and modeling. So we really focused on the PK components because that's foundational to understanding the PK/PD. If we don't understand the disposition of the drug and the exposure to the drug, then everything downstream is going to be difficult for us to quantitate and to optimize. So what you have to look forward to in the next session is an expansion beyond PK. First thing we're going to talk about is no PK and the disease progression and systems biology modeling that I spoke about earlier, which actually are understanding the disease without any intervention. Then we'll look at how the drug impacts disease progression, how the drug impacts pharmacodynamics on a rather macro level, and how the drug impacts pharmacodynamics on a micro level by talking about quantitative systems pharmacology. So I hope that this has been useful for you. Thank you so much for joining me. This is going to be now the end of part one, and with that, I invite you to please keep in touch if you have any questions, and please join me for part two.